does cell culture technology mean? It is the technology in which you actually culture cells. So first of all, you go to a store, you buy some cells, you plant it, and it's so important, don't forget to water it. It's not really like this. It's not culturing cells. You're not going to put cells in the ground and water it. Actually, it's a little bit more complicated than this. First of all, we're going to go step by step. It's a huge science, but step by step. First of all, we need cells, right? If we want to culture cells, so we need cells. From where are we going to get cells? From tissues, from our bodies. But I'm not going to get cells from my body and put it in a culture. So I'm going to isolate. I'm going to get a tissue, put some digestive enzymes in it. It will digest the proteins, and I might put something like EDTA to chelate the calcium, which connects the cells. And finally, I'm going to have my cells free, so I can take it and culture it. There's lots of types of cells. I can get my cells from plants, from insects. I have lots of famous insects. I have the Drosophila melanogaster.
Go on.
I have the Sporoptera, Frogibera. Sorry for the names, but I cannot keep them. And I have the Tricholosia. From its ovarian, I can get cells which is named hyphae. So now I have my cells. I want to culture it, so I put it in a container, and maybe I'll just wait for it to grow up. No, I actually need to add something. You know, a baby can't grow up to an adult if you didn't feed him, feed his soul, and feed his body. So now we're just going to talk about the body. So I have to feed my cells. I have to put them in a medium that they can grow in. So over years, medium has developed a lot. First of all, the first thing that a scientist would think of, I want to put a cell in a container, in a flask, in a petri dish, in anything, and I want to make it grow. So what is the first thing that will go to your mind if the first medium that you can put your cell in? I guess it's the original one, right? So like serum, we can put proteins. It's just like maybe blood or the things that are existed in the tissue, the extracellular matrix, and so on. So the first idea was this. They made medium contained animal products like serum, albumin, and growth factors. Then these products, these medias, had lots of advantages and disadvantages. So when you have albumin, proteins, growth factors, etc., there can be a lot of interactions with the cells. And my cells, maybe they're not going to grow the way I want. In addition, there might be contamination in viruses and bacteria and so on. So they thought that I have to get the additional things, like maybe I have to to eliminate the proteins, the serum. So they made animal products, free mediums, ABF. They first started with the plants hydrolyzated like soy pectin and yeast extracts. And finally they came to a CDM, which is chemically defined media. That means a media which is really defined, like I know every single component in it, and there's no extra things, there's nothing that will make contamination for me. So, the original components that can be in a medium, buffering system, inorganic salts. Inorganic is so important to keep the balance at the two sides of the membrane. So my cells are not going to explode and they're not going to shrink. Another thing is amino acids. It's the blocks for proteins. We all know. So we're going to talk about the goal of culturing. It's all about producing proteins. So I will need amino acids, carbohydrates for energy, proteins and peptides, albumin, transferrin, peptidoglycan. We said that we reached the chemically defined medium, but sometimes we still need those proteins. Fatty acids and lipids, vitamins, trace elements, media supplements, and antibiotic and antifungals. Some very famous medias are the RBMI media, which is Roswell Park Memorial Institute medium, and the MEM, which is Eagle Minimum Essential medium, the DMEM, which is Dolbicose Modified Eagle medium, IMDM, Iscobes Modified Dolbicose medium, and the nutrition mixture of F10 and F12. They are so common. They differ in their components, they differ in their uses. I think we don't have really to go in those details right now. So, first step is actually done. I isolate my cells, I put it in a flask or petri dish, and I put a medium in it. So now I have to make it proliferate, right? So I have to make it grow and proliferate. First of all, I need to know why I want to culture. So when I want my cell to proliferate, I need to know the end of it. What signals I have to give my culture, my cells? What really components I have to add? So I might produce medicines, maybe host for viruses, maybe vaccines, products for tissue replacement, as we're going to see in the applications at the end, and products for gene therapy. It's really useful. Actually, I need to know which cell line I'm going to do. So if I'm using, for example, mammalian cells, okay? So mammalian cells is better than bacteria's in that mammalian can grow into large. Like I can make it commercial, not just for laboratories and experiments. And I can do things that actually the bacteria can't do. In mammalian cells, I can do disulfide bound, I can do glycosylated, add protein with the sugar that's glycosylated, while in bacteria, I can't. So every line has its disadvantages and disadvantages. So my cells are not really going, we said that we need to give them signs to grow and signs to produce. So if my cell doesn't really normally produce insulin, so how can I produce insulin? I will do something which is called transient and stable expression. I'm going to add DNA, my DNA, the DNA of the insulin, to the cells and maybe incorporate it in the DNA of the cell, or maybe not. So if I incorporate it in the DNA of the cell, it's going to be stable expression, and if I didn't, if I make it just alone, a chromosome which is going to duplicate alone of the DNA of the cell, so it's transient expression. Everything has its advantages and disadvantages, as we say. Transient is going to be for small quantities, small, large, and the stable is going to be for large and commercial things. So now we have a video. It will explain how do we enter the DNA into the cells.
Transfection is the process of delivering nucleic acids into eukaryotic cells while minimizing unintended effects such as toxicity, off-target gene perturbation, or cell death. Because nucleic acids are hydrophilic molecules and the cell membrane is hydrophobic, coaxing DNA past the membrane can prove challenging. Transfection can be accomplished by...
So first of all, our DNA is hydrophobic, so we need to, hydrophilic. So we need to make it hydrophobic to enter, the membrane is hydrophobic. So they're going to talk about 3 ways.
A variety of techniques, such as lipid-based transfection, or transfection using calcium phosphate, or electroporation, the use of electricity to transport DNA into the cell.
So that's how are we going to enter our cells. There's 3 different, they said, and there's more. Now I have a cell, I put a medium. I transfected I transfected the DNA, I put it in a medium, I made it proliferate, so I'm going to have something which is called the cell line. Cell line is not really actually like cells are just in a line. It is a couple of, the result of the proliferation of the cells. I need to do something which is called screening the cell line. So lots of cells, I need to choose the ones I want and the desires I want. So that goes in many ways. Then I'm going to have something which is cell culture. And I can make my cell line proliferate to have a big cell culture, or I can do something else, which is freezing. So I can freeze my cells with nitrogen in -0 temperature, and with this cool and colorful bag, so it's very suitable for girls if you want, and guys can use it. It has alcohol, not nitrogen, alcohol, so I can freeze my cells in it. And then I can do something which is the opposite of freezing, which is thaw-freezing my cells, and then proliferate it again. Okay. That whole process needs equipment. First of all, we need cell culture hood. Cell culture hood, it's like a cabin where I do the culture, the whole thing from the cells, from the isolation to the cell culture. I will do it in this hood, which protects me from dust, from any contaminations can be in the air. And that's it. I need to do, I think you all know the horizontal and vertical flow. So, I think the best is the vertical one. That's the hood. It can be opened and not all of it. So let you just open it for here. This is very important. The ethanol 70% is very important. You know, every step you need to put it on your hands. You need to put it on the whole hood when you finish and when you start. So everything is just right here. The other thing is the incubator. We have actually two incubators. The first, the top one is to make the cells in 37 degrees. And the bottom one is to preserve the cells. Water bath to reach to 37 degrees for the cells. I need a centrifuge, refrigerator, and the freezer. We said we're gonna freeze our cells, right? Okay. I need a cell counter. I can go to the automatic one, or I think you all remember this, you know, in the third year, I think. Okay, it was good. I think you can go to the automatic one. Okay. So I need also inverted microscope. Inverted microscope is a microscope, but it's actually upside down. You know, the lens are just, you know, under, the light is above, everything is upside down. That is very much better from the usual microscope. Because it allows me to see my cells in a tissue container. You know, I can see in 3D. It's not just like the glass we put and we see from it. No, I can put a container and see the top of it, the bottom of it. Next, liquid nitrogen freezer for the freezing. And sterilizer, like autoclave. Expanded equipment, additional equipment. We might need those equipments that I'm gonna use it in my hands. You know, first of all, the aspiration pump. It has to be peristaltic or vacuum. You know, it's negative and positive pressure. That's the peristaltic. And sorry, that's the vacuum. That was the vacuum. Confocal microscope. Okay, sorry, I think this is going to give me the three-dimensional image, not the other one. pH meter, that's a portable one. Flow cytometer, which is going to provide the stream. Peptics and peptitorns. I think we can use it in our laboratories, actually, in the college. So much needed. Waste containers. No, every single thing has a single waste container. Like, no, they said here for autoclaved things only, and Biptix has one, and every single component has a separated container. Cell culture vessels. That's the flask where we're gonna culture our cells. Or we might culture it in petri dishes, and we can roll bottles, we can use it. And multi-well plates. That's very important for transcription. It gives a good transcription, so no many mistakes are going to be. Syringe and needles, media, and absolutely we need the cells. Aseptic technique. Everything we do in the cell culture needs to be aseptic. We're not going to clean anything like dust or something, but that's what we're going to use. This video explains actually everything we said. It's about taking the cells and changing the medium, everything, but in a aseptic way.
sterile technique, always wear basic personal protective equipment, or PPE, when working in the laboratory. Talk to the safety team at your institute for your required PPE. Also, remember to review the MSDS information before working with any media or reagents. Wear closed-toe shoes and clothes that cover your legs. Washing your hands before handling cells or performing culture work removes bacteria and microscopic dead skin particles. Dead skin cells can be a potential source of contamination. A 70% ethanol wash kills microorganisms that contaminate your cultures. Cleanliness is one of the most important parts of sterile technique. Always clean the hood before and after use. Spray alcohol should not be used in any area where a flame is being used due to the fire hazard. The outside of containers carry dust and contaminants. Remember to clean each item placed in the hood. The work surface. Not only does this increase the risk of contamination through accidental touching, but it also interrupts the airflow through the chamber, which will not maintain the sterile field. For this same reason, it is important to keep the front sash in the lower position when working. The basic idea is to keep your media and reagents sterile by only touching them with sterile objects. Using sterile media, reagents, and supplies is a big step in keeping your cultures free from contamination. Notice how the cell culture hood is set up to make the following actions easier. The pipette aid is on the right side, so you can easily control the pipetting in your right hand. Reagents are in the center back.
You know, they say exactly, like you have to put this on the right, you have to put this on the left. All of this to not have contamination in your body.
So you can easily open the bottles and pipette from them. It is very important to have the setup in this manner, so you're not crossing your hands or supplies over top of sterile items. We'll now demonstrate the basic technique of handling a pipette and a bottle of medium while aseptically supplementing Dima with Glutamax. When opening the pipette package, be sure the pipette does not touch anything non-sterile. Grasp the pipette high on the neck. Insert the pipette into the pipette aid. Turn the desired measurement marks toward you, and then discard the wrapper. Discard any pipette you accidentally contaminate. Only open your media.
Like everything is disposable. Everything is just used for one time, and then you just throw it.
Reagents and supplies in the cell culture hood. Opening these items outside the sterile field will result in contamination. When holding the cap, it is important not to touch the inside edge, or you could contaminate it. Replace the cap as soon as possible. If you must set the cap down in order to free your hand, set it down with the interior surface facing down. When pipetting, try not to touch the pipette to anything non-sterile, particularly the outside of containers, or contamination could result. Each pipette should be used one time and discarded, or washed if using glass pipettes. Reusing pipettes by dipping directly into another media bottle, or by leaving it standing in a media bottle, increases your chances of spreading contamination. Gently mix the contents after supplementation. Labeling the bottle after supplementation is good laboratory practice. Sometimes you'll need to transfer large volumes of liquid from one container to another. The best way to transfer is always to use a sterile pipette, but with practice, it is possible to aseptically pour instead. You should pour quickly and deliberately, using the front corner of the bottle to channel the liquid and improve the speed and accuracy of your pour. Any spills should be wiped with ethanol immediately. When passaging cells or changing their medium, the basics of aseptic technique are the same. Only open the containers in the sterile field. Do not cross your arms or other items over an open flask. Don't rush, but work at a good pace and with deliberate motions. Do not reuse pipettes, and do not use items that were inadvertently contaminated. When finished, make sure everything is closed tightly before removing from the cell culture hood. Anything opened outside the sterile field will now be non-sterile and should not be used for cell culture work. Wipe down the work surface with ethanol again and straighten up the hood before you leave.
See, every time like, you have to just put ethanol 70%. When you finish, when you start in the other way. So that's the aseptic technique. Now I have a cell line. I made it cell culture. So that Petri dish or that flask is really not limited, right? It's a space, a limited space. So my cells are gonna proliferate until there is no enough space in this flask or in this Petri dish. That's called that they reaches the confluency. So the confluency here is 100%. Confluency means it's a measure for the average of the number of cells that cover the our flask or Petri dish. Sorry. So when I say that confluency is 100%, that means there is no more space in the Petri dish or in the flask for cells to grow. Maybe I can say 50%, the confluence is 50%, so there is enough space. So when there is no enough space, what can I do? There's no enough space for cells to grow. So exactly. I have to move them to another Petri dish or another flask. This technique is called subculturing or passaging. So if I didn't do this, my cells are going to die, actually. There's a video also about passaging. It's all from no meant for trade names.
The growth of cells in culture follows a standard pattern. A lag after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate or the cell density exceeds the capacity of the medium.
So there's a bunch of time when there's no enough space for cells to grow. They will start to, there is no growing, and then it will just go down. There's that.
Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration.
So first of all, when we get the flask from the incubator, we have to make sure there's no change in color. We have to examine it under the microscope to show the morphology of the cells. So it didn't change, and my cells are in the right direction.
Aeration. Handle the cells with care during transport.
We have adherent cells and suspending cells.
For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution, such as DPBS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle cell dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless.
So that's under microscope.
Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Transport the flasks to the incubator and use a north-south, east-west motion to evenly distribute the cells.
That's the way of moving.
When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You will follow the same counting procedure for adherent cells using Trypan blue in either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells. Remove the medium and replace with completely new medium. This eliminates cell debris and metabolic waste.
So every 3 weeks, actually, you need to do that massaging. Okay, so all of what we're talking about is 2D. There's something new. It's called. It's not really new. It's from years. It's the 3D. So 2D cultures, the cells are just adhered or suspended in the medium, and the 3D, like we're gonna have preserve the shape of the cells. A video, please. I'm sorry for lots of videos, but there's lots of things that really can't be explained without imaging in videos.
Alvatech Scaffold, real 3D cell culture, delivering powerful improvements over conventional 2D cell culture. Tissues are made of highly complex 3D arrangements of cells. Maintaining the 3D structure of a cell is a key challenge in cell culture. Limitations of 2D cell culture. In conventional 2D cell culture, the structure of the cells changes as they grow. Cells distort and flatten, forming a simple monolayer. Up to 50% of each cell may be in contact with the medium. Up to 50% of each cell may be in contact with the plastic. The red line should...
So here I'm losing like 50% of the surface. 50% of my cell is not really contacting with the medium.
is the surface where no direct cell-to-cell contact is made. Cell-to-cell interactions are therefore virtually eliminated.
And even I'm losing the contact between the cells.
Alvatech Scaffold revolutionizes cell culture. A unique, highly porous polystyrene scaffold. permeate freely into the scaffold. Cells can form complex tissue-like 3D structures. This visualization illustrates the benefits of 3D. Confocal imaging of cell cytoskeleton structure. On the left, conventional 2D. And on the right, Albertex Scaffold 3D. Albertex Scaffold delivers more in vivo-like cell shape, cell-to-cell interactions, and cellular organization. Albertex Scaffold, enabling real 3D.
Again, no need for trademarks. It's just because that video was cool, so I needed to put it. Okay. So now we finished the first step, which is the cell culture, which is the most important thing. And now I need to make this larger. You know, I don't just need cell cultures in my laboratory. I need something to produce quantities, to produce larger quantities. So I'm going to go with something called bioreactors or fermenters. Actually, normally it is the same, but bioreactors are for the eukaryotic cells, and fermenters are for prokaryotic cells, right? So what is a bioreactor? A bioreactor actually has lots of types. It's classificated. According to lots of things, according to stability, according to homogeneity, according to something like it's large or small for suspending cells or adherent cells. There's lots of types, and when I really go, there's something called single-use bioreactor. Single-use bioreactor is a bioreactor that you use for one time, and then you just dispose it. You don't need it anymore. It's very important that you don't need any more sterilizing, no more cleaning. You will lower the cross-contamination, save your time from sterility needs time, so you're going to save your time. Less space for production, limited time for installation, and of course a better production yield. A commercial name is universal, just if someone looks for it. Aeration. So bioreactor has cells. Cells are living. So just like a human, the human needs aeration, needs oxygen, needs air. So we need to make aeration in the bioreactor. The main thing, the key point at sure, is the oxygen. So I have to really maintain the quantity of the oxygen which is in the bioreactor. Low oxygen is going to cause me problems in cell growth, and excessive oxygen is going to cause death, maybe. The most common aeration way is sparging. Sparging is including bubbling the gas into the liquid directly, and the other ways are like problematic. They have lots of problems. But a new study in 2014 says that we can get not the whole air in the bioreactor. We're just going to get extreme, which is pure oxygen. That was helpful. This didn't give me contamination that can go if I just enter the whole air with all of its components, even if I filtered it before. So I'm going to get a stream of oxygen and with filters. This is really amazing, and they are working on it now, but the problem is that oxygen, we know that oxygen might ignite some materials that are in the bioreactor, so they are working on passing it. Mixing. There is lots of ways to mix. Mixing is very useful in the bioreactor and must be in the bioreactor because you need to have the homogeneity, no suspending, no sedimentation, and you need the whole cells to get the whole media, you know. So there's the standard. That's the Rochen. The Rochen to. It's like horizontal or something. I don't know. Okay, the Rochen is turbines that go in the media, the whole media. So the media, like not like we have something sedimented and something flow and so on. So everything it's homogeneity. That's the standard tanks for mixing. The stirred tanks and the bottle arrangements.
Super.
So hydrodynamics. This mixing and every component in the bioreactor because hydrodynamics. Hydrodynamics are not really specified, and the reason exactly is not really known. But something will cause hydrodynamics is the microcarrier concentration. Microcarrier concentration. Microcarrier is a matrix that you suspend your medium in it, and kinematic viscosity, which is the viscosity on the density, and the agitation power. So low agitation won't cause problems with hydrodynamics, but high agitation will. And sometimes we need high agitation, so we need to pass this. Control, I have in my bioreactor to control the pH, the salt, oxygen, temperature, media delivery, and harvest. Salt separation, product capture. After I have my bioreactor is working, the cells are producing. So now I have to separate my product from those cells and get it. It goes in many ways. Just for the time, I'm not gonna go so much details. Downstream processing is how to, like when I get, for example, we said insulin, right? So when my bioreactor produces insulin, I could capture it, and when I capture it, I have to purify it. My protein would be contaminated in so much things. So that's called downstream processing. Actually, since they invented, or they discovered DNA, they just spoke about the upstream. Upstream is the things before the downstream, you know, is how to repair the cells. But really downstream must have it's very important because my protein might not fold correctly, might degrade. So I need to purify it and get it in the right shape. Formulation, filling, and packaging. So the end, I'm gonna get this, purify the protein, purify the product, and how I'm gonna get it to the patient if it's medicine. I need to actually, package it. I'm not gonna package it in a present or something, but I have to package it in I have first to choose the excipients I need, the route of administration. Am I gonna give it for my patient orally, intravenous, intramuscular, the route of administration, excipients, and the container and everything. It has to be perfectly, like when the patient's gonna get it, he's gonna get it in the final way. I want to say something. I forgot. Yeah. Okay. So it differs. Like if I want to get, I have to mention that it differs. If I'm gonna get it to my patient, like from the pharmacy or from the hospital, there's difference in route of administration and way of dealing. And sure, I have to be very careful for something that I am a company. Actually, trading is part, like beside the science, there's trading. So I'm a company. I can't like generate a product which is intravenous, which I can administrate intravenous, and there's another competitive company produce the same product which is orally. So now not really getting any useful. Same product, orally and IV, it's not useful for me. Okay? But if it's like more effective, so I can think about it. Okay. Now some applications of bioreactor, which is the most interesting thing. First of all, the production of viral vectors for genetic investigation. That's about mimicking the environment of the viral. We said the signal we put the medium, etc. And adult stem cell culture. It's very important that they actually invented 2 bioreactors, InFred and MiniFred, which provides, that's from Harvard, that provides airway cells or airway organs. So I can make airway organs right now. What is this useful? You know, transplanting, when I want to transplant organs. So there's lots of risks, lots of things to do. I have to maybe the patient with no immunity for lots of days, lots of weeks maybe. So in this way, I actually generate an organ, whole organ. I can generate it in bioreactors. That's true. That's not anymore a dream. And there's other ways to generate organs, but one of stem cells. Now they are working on mesenchymal stem cells, the human mesenchymal stem cells. It's a little bit difficult. There is lots of risk, but they think it will be a cool invention. We're gonna see. Hematopoietic stem cells. It helps me with cancer, especially, and lots of things. Novel bioreactors stimulate urinary bladder mechanical. Actually, we know the bladder has 2 types of cells, the urothelial cells and the smooth muscle cells. So, in a way, they put those cells in a ciliastic and in a rubber rings. They made a pressure in it that is similar to the pressure that the bladder has. So those cells are actually working as a bladder. I do like which can work in the space, even if there's no gravity and so on, and they can grow mammalian cells. Like when I searched for applications for bioreactors, I thought that all of the applications are just about alga. Like everyone was talking about alga. So I think it's the new invention now that alga can feed a house. That's a real house. It's not. That's real. It's in Germany. They made a whole building which is fed by alga. Like alga in bioreactors will provide the electricity, the water, and everything. It's all about alga. So the other use of alga in high school students, they are high school guys. High school students are making alga bioreactors. It's very, it looks old, it's not old, but they provide food. They want to produce from alga everything which is nutrients, and they want to give it to Africa, and they want to spread it in the schools. So that was my lecture. Thank you for listening. I hope it wasn't boring, and thank you. أولاً, can I talk in English or Arabic?
Whatever you want.
أول شيء بدي أشكر الدكتورة فوزة. عن جد دائما شو بعمل فيها مثلا دور ما دور عم يطلع معي. عن جد كان كثير حلو التعاون مع مشروع تخرجي. طبعا كنت مشروع يعني بس شكرا جزيلا.
واعطيتيني الفرصه عن جد لحتى يكون مشروع تخرجي له فعلا قيمه، يعني انا حسيت عن جد اني عم دور على شيء، حسيت انه اكثر شيء فعلا طلعت المشروع كامل، يعني حسيت انه خلص عرفت كل شيء عن الموضوع من اول لاخرته. فشكرا جزيلا
التقنية الحيوية قديش الموضوع شائك ومرق علينا بأكثر من سنة. نحن في مادة خاصة اسمها تقنيات حيوية ميكروبيولوجية، سبجكت كامل، مادة كاملة عم ندرسها بالسنة الثانية تبحث بهدول التكنولوجي، بالسيل كلتشر، بالبايورياكترز، بالكنترول اوف كلتشر، كيف بنتحكم بالبي اتش، كيف بنتحكم بالفوم، كيف بنتحكم بالايريشن، الهيدرودايناميك، لانه وهذا منطلب عملنا كصيادله، لانه انت بكره بدك انت تعمل بايو فارماسيوتيكال برودكت، والبايو فارماسيوتيكال برودكت هدول هلا سوقهم وهلا وقتهم، وهلا البورصه الدوائيه العالميه معتمده على البايو فارماسيوتيكال برودكت اللي هي كلها ادويه مثل جروث انسولين، ايكوبايوتين، المونوكلونال انتي بادي، كله بيعتمد على التكنولوجي. لذلك افردنا ماده خاصه اسمها تقنيات حيويه ميكروبيولوجيه، وهو المايكرو اورجانيزم هو البطل فيها، مثل ما وضحت او التيشو كلتشر. وبعدين هي احاطت، يعني انا كان قصدي لما اعطيتها الموضوع التيشو كلتشر بس هي لقيتها انه امتدت على البايو رياكتر، فكتت قلت لحالي يلا خليها تجمع لنا بمخطوطتها كل هذا الحكي، لانه البايو رياكتر يعني الحقيقه سيستم ثاني غير سيستم التيشو كلتشر، هي حبت تكمل الموضوع من اول السيل كلتشر. ومشكوره جبتي لنا اسماء الانسكت، الحشرات اللي كل اسماء اكبر منا للحشرات. وهذا كمان هلا شيء جديد عم يستعيدوا الماميليان سيل بالانسكت سيل للكلتشر، والحقيقه الامثله والصور تبعها مو شيء جديد على ديما انا بعرف انها كمان لها نشاطات كثيره اجتماعيه وثقافيه وكمبيوتريه، ما هيك؟ ويعني شامله،
ايكولوجيا واشارات الصم،
شامله جدا جدا.
مدرسه بالصم
كمان بتدرسي
تدخل لغه الاشاره. نعم. وعندها طلاب اخذوا تاسع ونجحوا في التاسع ونجحوا
بتعرفي
فتدخل لغه الاشاره 100%، ومدرسه طبعا تطوعا منها، طبعا بدون مقابل بدرس بالمعهد.
برافو عليكي، يعني كمان ناطقه وغير ناطقه.
ناطقه.
احسنتي. في حدا بيحب ينوه شيء من طلاب السنه الاولى تقاني؟ طلاب سنه اولى تقاني، فاتن، هانية، هبه، لانا.
She hooked us everything was perfect. She gave me thanks for her.
اوكي، يعطيكي العافيه، شكرا لك، وشكرا للصبايا والشباب اعطونا من وقتهم لانه اليوم درسهم هن، وقطعناه كمان جبنا لك الجماعه عن الستاج فوق بالمخبر اجوا وحضروا لك، ويعطيكي العافيه. يعطيكي العافيه. خلينا سيدي وبدنا نعمل نسخه للصبايا والشباب. سيدي الوورد غير نسختي، بدك تهديهم نسخه للسنه الثانيه مشان اكيد في شيء نحتاجه بنضيفه للمنهاج تبعنا.